Hi guys, today we're going to talk about the introduction of our food product, the fermented salted fish. Now first, we need to know where was this originated. Salted cured fish, popular as flavoring in local dishes in Senegal and West Africa, gutted or whole split fish are packed between layers of coarse sea salt and fermented for 1 to 3 days. Despite their popularity, research and development on fermented foods is meager. Most of the traditional food fermentations in the States, especially in the Philippines, are rural, seasonal, labor-intensive, informal, and capital deficient. Commonly, fermented foods are sold and consumed in areas where they are produced. Now on to our product. This product is popular tradition food in Central Luzon. It's usually prepared using freshwater fish. During fermentation, the fish flesh becomes very soft and the bones acquire the characteristic softness of a cartilage when cooked. Before serving, it is sauteed with oil, garlic, and onion. It is consumed either as an appetizer or a main dish.
Salting is the preservation of food using edible salt, otherwise known as sodium chloride. It is one of the oldest methods of preserving food. Salt prevents growth of microorganisms by drawing water out of the cells of microbes on the fish through osmosis. Usually, a 20% concentration of salt solution is enough to kill most unwanted bacteria. Salting is used because most microorganisms like bacteria and fungi cannot survive in an environment with such a high concentration of salt. Due to its hypertonic nature, any living cell will become dehydrated through osmosis and die, or become deactivated. This temporary deactivation is what preserves our fish. Ginamos undergoes lactic acid fermentation, which is a type of fermentation caused by lactic acid bacteria.
Salted fish, or ginamos, is mainly composed of vinegar, salt, and fish anchovies mixed into a single dish, which is expected to be eaten weeks after it was made. To determine the science behind such bizarre dish, we shall trace back to its procedures. Before the fermentation process, we coat the fish with salt. As we coat it, osmosis occurs all throughout. After salt curing the anchovies, we place them in a salt and vinegar mixture and placed in a container to be sealed tightly and kept on the dark for 3 weeks. The sealing and storing of the dish is an example of a lactic acid fermentation. In this process, lactic acid fermentation creates ATP, which is a form of energy animals need when no oxygen is present. It causes glycolysis to occur, wherein it breaks down glucose into two lactate molecules. Lactate and hydrogen then form into lactic acid. This organic acid serves as a good preservative to the dish, since it contains antimicrobial properties. It is also a good flavor enhancer.
Before proceeding with the steps, we should prepare the ingredients first, which are bulina or desired fish, minced garlic and ginger, sea salt, and vinegar. For the first step, wash the bulina with water twice. The second step is mix the fish with salt. After mixing with salt, rinse again with water. For the third step, in a separate container, place the salt and vinegar and mix them together until the salt dissolves. After the salt has dissolved, place the bulinaw into the mixture and then mix. After mixing, drain the bulinaw but put the vinegar mix in a separate container. After separating, place the minced ginger and half of the garlic into the bulinaw, then start mixing again. And for the last step, place the bulinaw in a jar, add the remaining garlic, and then pour in the vinegar mix. After going through all the steps, seal the jar and wait for 2 to 3 weeks.